Hello and hi everyone. So welcome to the lecture of this topic. In this topic, kita akan study on the immunoglobulin structure and function. So antara point atau subtopik yang akan dilihat adalah the basic structure of immunoglobulin, isotypes of immunoglobulin, the special structure and biological characteristic of 5 immunoglobulin, iaitu IgG, IgM, IgA, IgD dan juga IgE, and also kinetics of the antibody response dan juga immunoglobulin superfamily. So this is the objective that you should be able to learn by the end of this topic. For example, like an understanding to the characteristic of antigens dan juga antibody, state the function of the FAB dan juga FC portion of an antibody, state what is meant by the biological activity of an antibody, dan juga you can compare the structure of the, for example, like IgM dan juga secretary of IgA with IgG as well. Okay, nothing. So for next adalah immunoglobulins adalah glycoproteins that found in the serum portion of the blood, which is composed of 82 up to 96% of polypeptide dan juga 2% until 14% of the carbohydrate. Itu adalah the composition ataupun the structure, the molecule yang membina the antibody. Merupakan humoral branch of the immune system ataupun immune response, as we have learned previously. Kita ada two types of immune system, which is the innate ataupun the adaptive immune system or the humoral. Has 5 major classes, iaitu IgD, IgE, IgM, IgA dan juga IgA. So this is roughly the structure as illustrated by the scientist ataupun the researcher. Dia mempunyai variable dan juga constant region, V and C, V light, okay, and C light, walls untuk V heavy dan juga C heavy. Manakala hinge region adalah between 10 hingga 60 amino acids. And kalau kita lihat, this is the structure of the immunoglobulin. Again, if you look at the basic structure of the immunoglobulin, this is amino terminal end ataupun FAB or paratop, di mana this particular paratop adalah the sites untuk bind to any antigen. Okay, the antigen mempunyai, dia punya on the surface adalah epitope. So therefore this particular paratop is very important untuk map ataupun suits dengan the structure ataupun molecule structure of the epitopes of the antigen. Okay, terdapat exchange, iaitu terdiri daripada for IgG adalah gamma, IgM mu, IgA adalah alpha, IgD delta, dan manakala untuk IgE adalah epsilon for dia punya H chain. So slightly different ataupun berbeza bergantung kepada kumpulan mana. Then untuk dia punya structure, they have two identical heavy chains yang each carrying covalently attached oligosaccharide groups, di mana two identical or non-glycosylate light chain linked by disulfide bonds. Okay, macam kita belajar DNA, so dia punya structure molecular DNA juga di bridge oleh disulfide bonds. Mempunyai two light change, iaitu varieties kappa dan juga lambda, manakala light and heavy change are subdivided into variable constant region, dan pada masa yang sama this particular light and heavy also contain amino terminal in variable region, carboxyl terminal in constant region. Okay, untuk things to ponder, this is roughly the basic structure. If you guys want to draw the immunoglobulin ataupun you were asked to draw this particular immunoglobulin structure, so this is roughly how the idea untuk you tulis, and mestilah lengkap, including the antigen sites ataupun the paratop tadi, the constant region, variables, light change, hinge, dan juga di bawah ini adalah heavy change. So di heavy change tu mestilah ataupun yang berat tu mestilah berada di bawah, so that baru dah dia boleh hold yang di atas ataupun the structure of the atas. Kalau letak yang di bawah ini adalah light chain, so which is very difficult lah untuk hold, as we know that sesuatu yang berat mestilah boleh menampung yang ringan. Okay. Seterusnya again adalah proteolytic treatment of immunoglobulin, di mana untuk this particular treatment immunoglobulin mempunyai enzim papain yang akan cleavage the IG into 3 fragment, di mana of approximately the same size. Two of the fragments called fragment antigen binding atau FAB retain the ability to bind antigen, manakala the third fragment ataupun fragment crystallized or FC cannot bind with antigen but is responsible for biological effector function like complementization, binding to macrophages ataupun natural killer cells and also neutrophil. Manakala treatment with enzyme pepsin, okay, ada dua kat sini. Proteolytic treatment adalah ataupun di run by enzyme papain, manakala untuk treatment with enzyme pepsin pula results in a divalent fragment called FAB2, which consisting of two fragments that are joined by disulfide bonds dan juga several FC fragments. So this is how it looks like kita punya immunoglobulin, yang mana yang sebelum ini kita nampak mereka hanya mempunyai sort of like on the punya surface adalah paratop or the binding sites for the antigen, tetapi this is roughly the structure bila ditengok secara molekular. Okay, this is isotype of immunoglobulin. As I said, IgA, GD, E, G dan juga M, and then also the properties ataupun where we can found each of them. And also this one adalah special structure and biological characteristic of Ig. So as you can see that this is slightly different between those 5 groups, iaitu bagaimana bentuk IgM cell. This is IgM, IgG dan juga IgD, dan also IgA and IgE. And then kalau kita lihat, this is how it looks like for the dia punya structure molecule, iaitu IgG, IgD, IgE, manakala IgA adalah dalam bentuk dimer, manakala for the IgM adalah pentamer. So kita boleh lihatlah kalau penta berapakah dia punya hands untuk dia membentuk satu molekul. Okay, for IgG, most abundant in serum, about 80% of total serum. And also ada 4 subclass of IgG, iaitu IgG1, IgG2, IgG3 and IgG4. And selalunya bin transfer ataupun dipindahkan dari mother to child via placenta during the pregnancy. Manakala for IgM adalah dalam bentuk pentamer, monomer of the membrane dan juga pentamerine secretion. First antibody in neonates ataupun baby, dan juga first antibody in primary response. So inilah immunoglobulin yang berada dalam badan kita semasa kita masih bayi. Manakala for IgA adalah about 10 to 15% of total serum Ig dalam bentuk monomers, dimers ataupun trimer, and also tetramers in serum. Dan selalunya orang kata majority dalam for external secretion like breast milk, saliva, tears and mucus of the bronchial, genitourinary dan also digestive tract. So this is about the IgA. And for IgE, potent for biological activity, extremely low concentration in serum which mediates the immediate hyper. Dan for IgE adalah berfungsi ataupun sensitive reaction, especially those who had any allergic reaction or symptoms like hay fever, asthma, hives dan juga anaphylactic shock. So ini adalah penting dalam sebarang kesan daripada yang mengalami or people or person yang mengalami allergic reaction di dalam badan mereka. So this is how the IgE akan been accumulated untuk membantu melawan the process of allergic reaction. Manakala for IgD ada about 0.2% of total serum yang berada dalam badan kita. Then together with IgM is the major membrane, sebab IgM adalah about 80 to 96%, right? So normally it can bound to Ig or mature B cell, though towards the end the function untuk activation of the B cell whilst no biological effector function has been identified so far for the IgG. Okay, this table shows you the change composition of the 5 immunoglobulin classes in human. So just for your information, we have 5 classes of the immunoglobulin. And if we look at this one as the kinetic of the antibody response, so kita dapat lihat days post injection after the first infection of any antigen, and then bila kita dapat the second reinfection dari the same antigen, so what will happen to our antibody? So the production akan been released ataupun reaction tu akan less time compared to the first infection, sebab pada masa yang sama our body system also telah pun mempunyai the memory B cells yang akan wait ataupun for example like standby for any ataupun the second reinfection by the same microbes or antigen. Manakala Immunoglobulin Superfamily is a large protein superfamily of cell surface dan juga soluble proteins that are involved in the recognition, binding dan juga adhesion processes of the cells. So as we can see that this is the superfamily of the immunoglobulin, as IgG, TCR, CD8, CD4, HLA class 1 dan juga HLA class 2. Manakala some CAM adalah calcium 2+ dependent, manakala some others are independent. Okay, contohnya adalah cadherin, selectin dan juga integrin. As you can see that this is the component. Manakala as you can see that down here merupakan Ig superfamily. So the lots of very small and tiny components, they are helping each other ataupun membentuk immunoglobulin superfamily. The superfamily, kalau kita lihat, this is the example. The punya molecules have a series of globular Ig-like domains formed by disulfide bonds, manakala dia akan mediated CA independent cell adhesion, dan juga pada masa yang sama akan activate intracellular signaling pathway, dan the most important thing adalah play critical role during morphogenesis dan juga differentiation of the muscle, glial and also the nerve cell. And not forgotten, di dalam neuron, they promotes the formation of the myelin. So this is all very important dalam memastikan kita punya nerve system works very well. As you can see that here adalah immunoglobulin superfamily receptor. So we can see that ada IL-37, IL-2, for example IL-1 alpha, IL-1 beta and also IL-1RA, and not forgotten IL-33. So dalam immunoglobulin, this is the superfamily receptor yang akan or act sebagai untuk receive anything from the outside ataupun the foreign molecules. Okay, notes. Antibodies generally do not kill or remove pathogens solely by binding to them, but they are with the help from other cells inside our body, like neutrophil or macrophages. Then that particular antigen ataupun foreigners or foreign materials akan get rid from our body system. Untuk opsonization, the process, this is how it looks like. Di mana adalah the coating of a pathogen with antibodies that makes it susceptible to phagocytosis. Okay, for example, like this is the pathogens, and then the opsonization akan berlaku seterusnya. So this is how it looks like, di mana phagocytosis is the process of cell phagocytes engulf, ingest dan juga destroying the pathogen. So we can see that this is the molecule that outside the very light yellow yang akan engulf ataupun covered this particular pathogen, and then di mana the neonates ataupun the baby have a decreased amount of opsonins, iaitu antibodies that promote opsonization. So sebab kerana baby adalah baru saja detached from the mom, so they are developing and akan takes time untuk develop dia punya own antibody. Manakala di sini adalah kalau kita lihat how the process takes place, sama juga macam tadi. So secara roughly bagaimana antibodies akan bind to the antigens, and also stop their destructive behavior in one of several ways. So as you can see that this is how it looks like untuk antibody bekerja yang akan bind to the antigen. So like said previously, this is the macrophage that we have inside our body system. Dan the opsonization akan berlaku where the antibodies akan engulf ataupun the opsonin akan engulf that particular pathogen, and then towards the end these particular molecules ataupun the binding molecules akan be ingested by the macrophages. So the macrophage akan starts to release a lysozyme untuk ataupun destruct the molecules of that particular pathogen, and then which later discards from our system. Okay, and this one also tells you give you an idea how the process takes place. Okay, manakala untuk activation of complement C1, we have the antibody here, we have the fragment C, and also with the help from the C1, R2 and also S2, this is how it looks like kalau untuk that particular antibody akan berfungsi terhadap this particular antigen. So towards the end, this particular antigen dia akan bind with the antibody akan works on this particular antigen, and then towards the end dia akan dibawa masuk ke dalam inside our cells, and then seterusnya akan being destructed. So dalam the immune system terdapat 3 pathways iaitu alternative pathway, lectin pathway dan juga classical pathway yang akan berfungsi untuk memastikan those particular antigens or the aliens that get into our body can be get rid ataupun discard out from our system. So as you can see that for alternative pathway, the pathogen surface create local environment conducive to complement activation. And then this is the first act, manakala the lectin pathway adalah the mannose binding lectin binds to pathogen surfaces, as the second act manakala for the classical pathway adalah C-reactive protein or antibody binds to specific antigen on the pathogen surfaces. So all these 3 pathways actually they are completing each other, so that's why we name it as a complement activation. They need or we need one to another. Manakala after this, after the complement activation completed, so the cleavage of C3 to C3a and C3b covalently bound to surface components of that particular pathogens, and then akan melalui another 3 processes like recruitment of inflammatory cells, opsonization of pathogens, facilitating uptake and killing by phagocytes, dan juga perforation of pathogen cells membrane. So towards the end, this is how the process which later takes place, and then dari proses inilah akan the pathogens akan dihapuskan ataupun dimatikan, or akhirnya akan di-lyse by our cell. Okay. And for this one adalah antibody-dependent cell-mediated cytotoxicity or ADCC. As you can see that we have the combination of the neutrophil, the NK cells, we have the target cell. This is the target cell, meaning that this is the foreign molecule or antigen, and also we have the eosinophil, and also then not forgotten the macrophages. Okay, so they are working together in order to target ataupun untuk get rid that particular target cell atau the antigens dan yang get into our body system. They are not alone. And manakala this one adalah for antibody transport through epithelium or placenta, dipanggil transcytosis, di mana the movement of antibody across epithelial layer. Okay, so this is the epithelial layer. Dan also how the delivery of IgA to the mucosal surfaces of the respiratory, gastrointestinal dan juga urogenital tracts, as well as its exports to breast milk, di mana the IgE being transferred from mother to fetus melalui passive immunization akan berlaku. So kita dapat lihat di sini bagaimana on the surface of the epithelial layer akan cross to make sure that, and also pada masa yang sama, this is the antibody on the surface of the epithelial layer. For the activation of mast cell, eosinophil dan juga basophil by IgE, okay, di mana terdapat we name it as immune phase 2 melibatkan mast cell dan juga eosinophil and basophil, where antibodies bind to the surface of mast cells, basophil and eosinophil, and which later cause release of cell content that leads to the reaction. So what will happen to us, sometimes bila berlakunya reaction, those reaction inside the body, so towards the end, on top of that, so reaction sebagai contohnya, if we not feeling well, the flu, so normally kita ada fever, for example, itu adalah antara cause reaction of hasil daripada tindakan-tindakan sel di dalam badan kita. Okay, the first immune phase of the allergic reaction, kalau kita tengok, this is how it looks like, ataupun bagaimana berlakunya kalau allergic reaction. Kita ada mast cells, kita ada basophil, and kita ada high affinity of IgE receptor. Again, remember that the immunoglobulin that involved in the allergic reaction adalah IgE. For this one adalah IgA in the gut. Okay, what will happen? So as you can see that this is how it looks like bila IgA berada di dalam gut, and then this is dia punya function in the gut. So kita ada mucosal epithelia. As you can see that this is the antigen, and which finally akan been attracted ataupun dibawa atau engulf into the cell, where di dalam badan kita adalah organized lymphoid follicle, which later we have the plasma cell inside this particular lamina propria ataupun di kita punya dalam badan sel, and the plasma cell akan functioning sebagai when they get notified, there's an antigen outside ataupun within the lumen. So the plasma cell get notified ataupun the signal, and then which later travel out from this particular mucosal epithelia, which later akan release the antibody untuk track ataupun trap or attack this particular antigen. Di mana secretary, kalau kita tengok formation of secretary of IgA pula adalah especially in breast milk, di mana binds to microbes in baby's digestive tract and thereby prevent their attachment to the wall of the gut and also their subsequent passage into the body's tissue. Manakala here adalah allergen, cross-linkage of receptor-bound IgE on mast cells induces degranulation. So kita dapat lihat bagaimana the allergen reaction berlaku di sini, and also the IgE akan being produced in lots of numbers, manakala the FC ataupun fragment C of the particular antibody tadi receptor specific for IgE. So we have the mast cell yang akan berfungsi ataupun menghasilkan IgE untuk react towards any allergic reaction. Dan degranulation, bila ada binding of the allergen dan juga IgE, so degranulation and release of granules content akan berlaku, and then the histamine, which later the histamine and other substances that mediate allergic reaction also akan be released. So towards the end, as I said, it's either if you are allergic to pollen ataupun the bunga or even the seafood, so normally after ataupun post reaction of from this particular reaction adalah kita akan dapat rashes or gatal-gatal, ada yang dapat sort of like become redness on that particular area. And IgE binds with high affinity to receptors on mast cell, basophil dan juga activated eosinophil. Longer half-life when cell-bound, initiate a strong inflammatory reaction to parasite especially, and also the paling penting adalah involved in allergic reaction. Manakala for IgD adalah antigen receptor on mature B cell, but no further function has been identified so far. Okay, this is the table showing that the activity levels, for example, whether it's high, moderate ataupun minimal or none or questionable for the properties and biological activities of classes and subclasses of human serum immunoglobulin. Okay, just for your Manakala this one adalah antigenic determinants on immunoglobulin, terdapat 3 iaitu isotopic determinant, allotypic determinant dan idiotypic determinant. Isotypic determinant adalah a constant region determinant that collectively define each H chain class and subclasses and each L chain type and subtypes within the species. So kita ada for example like here adalah mouse IgG1 dan juga mouse IgM. For allotypic determinants adalah differences in amino acid in C region. Okay, tadi yang H, which occur in some but not all members of a species, whilst for idiotypic determinants adalah the unique amino acid sequence of the V regions of a given antibody, whilst in some cases an idiotype is the actual antigen binding site. So as you can see that this is the B cell receptor, which is this is in terms of molecular structure. So just for your kind of information, don't worry much about it. Okay, for the antibodies terdapat 2 iaitu polyclonal dan juga monoclonal. For polyclonal adalah derived from different B lymphocyte cell line, manakala monoclonal derived from a single B cell mono. Batch to batch variation for polyclonal affecting antibody reactivity dan juga treatment, manakala for monoclonal no batch to batch variation, so very effective sebab they derive from a single B cell clone. Polyclonal not powerful tools for clinical diagnostic tests, dan for monoclonal adalah enable the development of a secure immunoassay system. Okay, this is how it looks like for the polyclonal antibody, dan this is how it looks like monoclonal antibody. Kalau kita lihat, for polyclonal antibody, we need more than one antibody untuk react on this particular antigen. Padahal for monoclonal antibody, hanya memerlukan 1 ataupun 2 antibodies. Okay, sebab they are made by identical immune cells and then clones of a unique parent cell. Okay, but and also in contrast to polyclonal antibodies. Again, this is the picture shows you the polyclonal versus monoclonal antibodies taken from the isolated serum from a lab mouse. Okay, and this is how it looks like, dan juga bagaimana the production of the polyclonal and monoclonal antibodies. Another slide shows you the production of the monoclonal antibodies. You take a serum from or isolated from a lab mouse. Okay, and this is the figures shows you the process macam mana kita inject the antigen to this particular lab mouse, and then we are taking the serum ataupun the serum or taking out some organ, especially for this one adalah spleen, and then akan suspend the spleen cell, so until kita dapat the hybrid cell and also the hybridoma dan juga desired monoclonal antibodies akan be produced. And also not forgotten, culture myeloma cells iaitu cancerous B cell akan di-mix up with the suspension of myeloma cell dalam suspension of the cells dari mouse yang di-injected with an antigen before that. Okay, so this is the evolution of monoclonal antibodies. Bermula daripada murine antibodies 100% menggunakan mouse. Bila we move to chimeric antibodies, so we just use 25% of mouse, dan the third one adalah humanized antibody, we just use 10% of mouse, and then the fourth one adalah the evolution of fully human antibody, which we use 100% from the human. For example, like adalim Mubat D2E7, and as you can see that from Cancer Research UK, and this is how it looks like of the cancer cell, and then how the monoclonal antibodies being used to treat the cancer cells. Okay, where the monoclonal antibody akan lock onto the protein of this particular cancer cells, and which later they are trying, or this particular monoclonal antibody akan try to kill this particular cancer cell. As you can see that this is all the very colourful one adalah the proteins on the surface of the cancer. Okay, manakala side effects of monoclonal antibody, in general there are more common side effects caused by monoclonal antibody drugs, including allergic reaction, hive or itching, flu-like symptoms including chill, fatigue dan juga muscle ache dan also pain, nausea, diarrhea and also skin rash. Okay, for example like here kalau kita tengok pada B untuk diphtheria toxin, and this is how the engulfment ataupun endocytosis akan berlaku, which later followed by endosome and the release of toxin into cytosol. So this is how it looks like for the diphtheria toxin, manakala this is what will happen to the immunotoxin, the tumor specific antigens, and then which later towards the end akan be released out from the body system. Okay, untuk polyclonal antibody response, as you can see that this is how it looks like. The antigen akan be injected into the mouse, lab mouse especially. So later the researchers akan dapatkan the serum from this particular mouse, and then they are going to develop ataupun bangunkan the polyclonal antibodies. Okay. For memory recall, okay, this is the structure ataupun 5 groups of the immunoglobulin that been learned in this topic. Okay, G, M, A, D dan juga E. And this is one of the example of the exercises that you can try at home. Let's pump up your brain for the antibody spread this question. So please have a look and then try to do it for yourself. And for the further reading, this is what been supplied for you guys, and then please read it as much as you could. Okay, try to make it as simple or in your own notes what is all about the immunoglobulin ataupun the antibodies that been learned just now.